About 3 months ago, I was invited to give a presentation at RADFest 2022 in San Diego. Now, I know a couple of you were there and saw that presentation, such as it was. Today, I decided to put up that presentation for everyone else. It's on the work of Dr. Michael Levin, and he's been doing some pretty amazing stuff in his lab, experiments that might possibly pave the way for the complete rejuvenation of human beings. Maybe. Before I get started, I've got to tell you a story about this presentation. When I got this invite, I was super excited. I've been to RADFest several times and I've made friends, and since this was the first time that it's been held since the pandemic, I was really looking forward to not only giving the presentation and being on the main stage, but also to reconnecting with a lot of friends that I had made there. RADFest happened the week after my son's wedding in Toronto, so I flew directly from Toronto to San Diego. The day before RADFest began, I learned that several people from the wedding party had come down with COVID. Now, thankfully, they're all just fine. But I thought that I should get tested nonetheless. Well, I tested positive, and I spent the entire conference isolating in my hotel room. Now, I did get to give the presentation, but virtually. I didn't get any feedback, I didn't get to reconnect with any of my friends, and most disappointing of all, I didn't get to hobnob with all the other speakers. All right, as disappointing as my presentation might have been, it's always been my intention to put it up on this channel. So here it is. Now, I'll skip the intro slide where I talk about who I am and my YouTube channel. I figure you guys already know all that. I started out by talking about my current interest in regenerative medicine and about some of the work that's going on in a lab that's studying regenerative processes back on the East Coast. But their work involves the regeneration of entire limbs or whole bodies. For example, if you cut the leg or the tail off a salamander, it will regrow or regenerate a new one. Planaria are tiny worms that can regenerate a whole body if you cut one in half. In fact, you can cut a planarian into 200 separate pieces, and each piece will regenerate a perfect new worm. The work that I'll be discussing took place in a lab at Tufts University in Boston, and it was headed up by Dr. Michael Levin. And they did something in his lab that I don't think has ever been done before. They regenerated the limbs from animals that don't have any regenerative abilities, like frogs, and coaxed these animals into regrowing new limbs. And they did it with very little intervention from the researchers. They learned a lot from these experiments, and some of the stuff they learned has huge implications for humans. So huge that I think it's kind of revolutionary. Okay, so one of the first things that they discovered is that the process of regeneration seems to follow the same pathways as the developmental processes, the processes that are responsible for turning an embryo into an adult version of the species. They're one and the same. The developmental processes that enable you to progress from an infant into a fully grown adult are identical to the regenerative processes that allow you to heal after an injury. It's all about making gradual changes to the structure in a series of small steps, progressively altering it over time. Now, when we're growing up from a developmental perspective, each prior stage of development is actually seen as a defect, as something that needs to be corrected. And our bodies make these corrections through a series of small remodeling events. These remodeling events have a couple of requirements. First, they need a snapshot of what the current state of the structure actually is, and then they need a snapshot of what they're shooting for, a target state, so that they can tell what the difference is between the two. And then these remodeling events simply try to reduce that difference, to correct the current state until it's the same as the target state. But as remodeling moves closer to the target moves on, so that we're always playing catch up. We're shooting at a target that is constantly advancing, becoming more and more mature until finally that target reaches its ultimate set point, our adult self. And then the developmental process shuts down. For humans, that's somewhere in our late teens or early 20s. Okay, so this remodeling process, this developmental process, it requires two things to function properly. It needs some sort of a mechanism for coordinating the cells so that they all work together towards a common goal. And it needs a stored set point, or snapshot of the target that the cells are trying to achieve. Okay, let's talk about this snapshot, this stored set point. This snapshot is called a morphological target, with morphology referring to the structure of things. So this morphological target is a state that we're trying to achieve structurally. Since the remodeling events are trying to correct defects, the snapshot needs to be error-free, a version of our adult self that is structurally perfect, right down to the cellular level. All of our organs and bones, our muscles and tissues, all of our cells, even our molecules, exactly as they should be. A state of anatomical homeostasis, where everything is running tickety-boo. So where's this morphological target, this set point that we need to be able to refer to? Where's it stored? In our DNA? Our DNA is a blueprint for our bodies, right? Well, turns out that idea isn't exactly correct. Our DNA is a series of genes, which is a section of DNA. When a gene is expressed or turned on, it expresses a protein. It builds a protein molecule. Now, how these proteins get assembled, that's not really stored in the DNA. And Dr. Levin proved that in his lab. They did an experiment on planarian worms, which are complex creatures. They have a brain, organs, a nervous and vascular system, and as I mentioned earlier, they have amazing regenerative capabilities. In this experiment, they cut the heads and the tails off of the planarians, leaving just the middle section. And then they reprogrammed the wounds from the amputations so that each wound regrew a new head. And they wound up with two-headed worms. Now, this reprogramming did not happen at the genetic level. All of these planarians still possessed wild-type genetics, just like any other worm. Now, here's what's interesting. If these two-headed worms still possess the genetics for a one-headed worm, when you cut them in half, you would expect them to revert to a one-headed worm, right? That each head would grow a new tail. But that's not what happened. Each half regrew a two-headed worm, and they continued to regrow two-headed worms consistently, generation after generation after generation, even though they all still possess the DNA for a one-headed worm. So Dr. Levin demonstrated that the morphological target that regeneration relies upon is not stored in the DNA. If it was, then the worms would have reverted to one-headed planarians. So if this morphological target isn't stored in the DNA, then where is it stored? Turns out it's stored in a bioelectric layer that sits on top of our genetics. The DNA expresses the organic molecules that we're built out of, and this bioelectric layer figures out how to assemble them to achieve the target that it's been programmed to achieve. So let's talk for a minute about bioelectricity. We all know what that is, right? It's what makes the brain and the nervous system work. Neurons create bioelectric potential, and this bioelectricity enables signals to be sent across our synapses. But all cells possess this capability. All cells have bioelectric potential. Neuroscientists have created tools to help them with their work on neurons. But these tools work equally well on every cell in our body. They can't distinguish between neuronal cells and non-neuronal cells. So every cell in our body is part of a bioelectrical network. And biology realized a long time ago that these bioelectrical networks were great for solving problems and achieving goals, and for storing information. Evolution discovered very early on that bioelectric signaling is a convenient medium for computation and global decision making. In fact, this capability rose with bacterial biofilms about 3.5 billion years ago. And this ability to solve problems through global decision making, as well as the ability to store information, is a type of intelligence. A primitive type, to be sure, but intelligence nonetheless. Okay, so what is intelligence? And what does it have to do with regeneration? According to Dr. Levin, the degree of intelligence is how effectively a goal is pursued. A higher intelligence can solve a goal more efficiently and more effectively. Cognitive sophistication impacts the size of the goals. An intelligence that possesses a higher degree of cognitive sophistication goes after bigger goals, like how to get to the moon instead of just where the next meal is coming from. Even a single-celled organism exhibits a primitive form of intelligence. It can solve problems and it can achieve goals. For example, it can choose to move to a location where nutrients are more available. But here's the thing. Once we evolved into multi-celled organisms, something very special happened. One of the most amazing aspects of multicellularity is the ability of a collective of cells to work together to achieve large-scale goals. A network of cells is more cognitively sophisticated than a single cell, and they can achieve goals that single cells just can't. And this is because of the bioelectrical network. Cells are computationally bound together. They can signal each other, they can communicate. They're bound into a collective intelligence. And all this happens because of the bioelectrical networks. This collective intelligence that has arisen within the bioelectrical networks in our bodies, it's this collective intelligence that coordinates and controls the cells during the developmental processes. But it's not just that. It's also where the morphological target is stored, the set point, the snapshot of anatomical homeostasis. Remember that the developmental processes require two things, right? A mechanism for coordinating the cells towards a common goal and a stored morphological target. It's in this bioelectrical network that both of these requirements exist. The network provides the control structure that is the mechanism for coordinating cells. And it's in this network that the morphological target is stored, not the DNA. All right, now that we kind of understand how both the developmental and the regenerative processes function, let's take a look at the work that Dr. Levin has been doing in his lab, and it's pretty remarkable. They've been investigating not only how the regeneration process works, but how they can tweak it to make it work differently. They've been experimenting with programming the bioelectrical network to produce different regenerative results. Using some of the neuroscience tools, they can observe the bioelectric signaling that goes on during regeneration. With programming, they can alter the signaling, which in turn alters the final results. And one of those results is the ability to coax animals without any regenerative abilities into regenerating amputated limbs. They've amputated the legs from frogs, and by reprogramming the bioelectrical networks that control the regenerative process, they've coaxed the frogs into regrowing a new leg. They've used neurological tools to read and write electrical information about morphogenesis. And they've learned a few things in the process. That's how they learned that the target morphology, the patterns of anatomical homeostasis, are stored in the bioelectric networks of multicelled animals and not within the genome. And they've learned that this target morphology is reprogrammable. It can be rewritten. It can be changed. They've learned that while no individual cell knows what the limb is or what it's supposed to look like, once the programming is finished, the collective can grow that limb. In regenerating new limbs, the cells work together towards a common goal, as established by the programming. And once the morphological target is achieved, regeneration shuts down. But here's what's really interesting. It takes about 1.5 years for a frog to regrow a leg that's been removed. But Dr. Levin's team did not have to micromanage the whole process along the way. They didn't have to tell the cells which proteins to build or how to assemble them. They didn't have to tell them how long the process should take or when to shut down the process. They also didn't specify which limb should be grown or what it should look like. They didn't do any of this. All they did was to trigger the regenerative process. All they did was start it up by sending a grow what's supposed to be here signal. Then they stepped back and let the animal do all the work. Because the animals already knew at some level how to build a new limb. The animal had already done that once before, right? The frog had grown the original leg that was amputated. So all they had to do was send a signal that triggered the regeneration process and then let the frog do what it already knew how to do. Here's the thing. you don't need to understand the repair process to get it to work. You just need to know how to trigger it. The system knows what it's supposed to do, and it has a control structure in place to coordinate the regeneration process. Now, very low in the control structure, you do need to tell it what to do. You need to micromanage every step in the process. But the higher up you go in the control structure, the more the system delegates the tasks, until you get to a point where all you have to do is to initiate the process and then just let it run on its own. So while the regeneration process took about 1.5 years, the triggering process only took about 24 hours. They used a device that they created called a biodome. It's basically a small sac or envelope that contains a fluid that's similar to amniotic fluid. By exposing the wound left by amputation to this fluid for 24 hours, they were able to reprogram the wound from simply scarring over into growing a new limb. Now, this next part is important, and it gives us a clue about how all of this relates to longevity. One of the potential perils to research into regeneration is the risk of cancer. Several of the regenerative interventions that have been tested have resulted in a higher risk of cancer. So if regeneration equals a higher risk of cancer, then you'd expect animals that have high regeneration potential to be more likely to develop cancers, right? But they don't. In fact, they actually have a much lower rate of cancer. And when these animals do develop a cancer, if you trigger the regenerative process while they have the cancer, they normalize all the cancer cells and clear out the cancer. Now, this is remarkable because it seems to indicate that during the regenerative process, it's not just the cells in the new tissue that's being affected. It's every cell in the body. Every cell is compared to a morphological target, and remodeling events make corrections until the tissue is the same as the snapshot. It's a global effect, and this holds some remarkable potential for reversing the aging process. Now, one obvious result could be that we're finally able to regrow and replace organs that are failing as we grow older. But there's another possibility, and I think this is really exciting. Let's consider when we start aging. We've been moving towards an ideal morphological target during the entire developmental process. But once we finally get there, the developmental process shuts down. We're no longer striving to attain morphological perfection, and all those small remodeling events are no longer happening. Instead, we're now moving away from anatomical homeostasis. We're getting further and further from that morphological target. Repair processes begin to decline, so repairs aren't as successful, or things get repaired incorrectly, or they don't get repaired at all. And so damage begins to accumulate. It's not much at first, so we don't really notice it, but as it accumulates, it becomes more and more apparent, until finally, in our 50s or 60s, we suddenly don't recognize ourselves in the mirror anymore. But what if we could trigger the developmental processes to fire up again? Not as developmental processes per se, but as regenerative processes, since the developmental and the regenerative processes are virtually the same. What if we could undergo a procedure that might only take a day or two, fire up all the repair processes, and then for the next year or two, or even the next 10 years, we slowly regain everything that we've lost. We grow younger, year by passing year, until we reach a morphological state that is similar to the one that we had in our early 20s. If Dr. Levin can regrow amputated frog legs over 1.5 years by simply reprogramming the bioelectrical network over the period of a day, will he eventually be able to do the same things in humans using a similar process? Might it be possible to trigger the regenerative process by maybe removing the last joint of our finger and then applying a biodome for a day or two? And if the regenerative process is not local, affecting just our fingers, but global and affects every cell in our bodies, might this be a path to complete regeneration of these aging vessels that we live in? Might it be possible to trigger a series of small remodeling events that gradually move us closer and closer to that ideal morphological target that still resides somewhere in our bioelectrical network, that snapshot of us in our late teens or early 20s? Now, I have no idea if this is possible or not. And if it is, it's something that you'd have to do over and over on a regular basis, because once we reach the target state, the whole process would shut down and we start aging again. But the advances in Dr. Levin's lab are very real. They definitely have been able to regenerate limbs and organs in animals with no regenerative abilities. And in the future, they hope to be able to achieve the same results in humans. But that's all a long ways off. And my speculations about the possible implications for aging are exactly that, speculations. Now, I have talked about this with Dr. Levin, and he said that nothing here looked too impossible. His words. But here's the point. It demonstrates that some of the research into longevity that's going on right now is way out there on the bleeding edge of what's possible. The discoveries that are being made, what we're learning about aging, it leaves no doubt in my mind that somewhere, someone is going to crack the code, and fairly soon. And it might just be Dr. Levin and his team at Tufts University. If you enjoyed this video, let me know in the comments below. If you'd like to learn more about the aging process and longevity, then check out this playlist on the hallmarks of aging. That's it for now. Catch you next time.